In this tutorial, we will learn about fluorescent in situ hybridization, or FISH. FISH is a technique that uses molecular DNA probes to detect complementary sequences along a chromosome. Unlike whole genome analyses such as karyotyping or ARRAY-CGH, FISH uses DNA-specific probes and will provide information both about the copy number of a chromosome region, so that's whether a region is deleted or duplicated, but also about the locational position of that region. Let's spend a bit of time thinking about how FISH works. A FISH probe is designed which is complementary to the strand of DNA which you want to identify. The probe is labelled or tagged with a coloured fluorochrome. The chromosomes of the target cells are arrested in metaphase, and then the cells and the chromosomes are fixed onto the surface of a glass slide. The slide is treated so that the genomic DNA is denatured into single strands. The slide is then washed with the single-strand probe DNA, and the probe anneals or hybridizes to the single-stranded target DNA, which is still in its natural position on the chromosome. That's a really key feature of FISH. So it is looking at the target DNA in situ. And because the probe is being tagged with the fluorochrome, which is a molecule which fluoresces when it is excited by a particular wavelength of light, it can be visualized using a fluorescent microscope. And indeed, several different probes can be hybridized, observed, and then compared simultaneously by using different colors and combinations of these fluorochromes for each probe. There are four major groups of FISH probes which anneal to different types of DNA. Alpha satellite probes are complementary to the repetitive DNA sequences found at the centromere. Although there is considerable overlap between the DNA content at the centromere, there's also usually enough differences between chromosomes that individual chromosomes can be distinguished. However, that's not true for chromosomes 13 and 21, and it's also very difficult to distinguish chromosome 14 from chromosome 22 with an alpha satellite probe. And as is often the case, it's actually usually really important to distinguish chromosomes 13 and 21. For instance, if a trisomy is suspected prenatally. And so in this situation, it's important to use other more specific probes that are specific to that chromosome region. Beta satellite probes are designed to anneal to the non-transcribed sequences of the acrocentric chromosomes, and these are chromosomes 13, 14, 15, 21 and 22. Beta satellite probes might be used to interrogate, for instance, extra material on the short arm of an acrocentric chromosome. Unique sequence probes are designed to anneal to specific chromosome regions, and these probes identify sequences usually present in only one copy number per chromosome. Unique sequence probes are therefore locus-specific and can detect a specific chromosome region or gene. And then finally, whole chromosome paints consist of a number of probes which cover the whole of the chromosome other than the centromeric region and contain a mixture of overlapping probes. And this is a picture of a whole chromosome paint, and you'll see that it will paint the entire length of a chromosome and can be used to identify the origin of unidentified material or rearranged chromosomes or marker chromosomes. So let's think a bit about the applications of FISH. Historically, FISH was used in a wide variety of applications, many of which are listed here on this slide. However, the technique is time-consuming, costly and laborious, and in many areas it's now been superseded by newer, faster techniques. For instance, prenatal aneuploidy screening is now commonly done using qFPCR, and ARRAY-CGH has superseded FISH as the go-to test for microdeletion and telomere screening. Nevertheless, FISH still has a role in confirming ARRAY-CGH findings, and it can be used to clarify the nature of a balanced and unbalanced translocation. It is found in an increasing variety of applications in hemato-oncology. Let's have a quick look at how FISH was used for aneuploidy screening, because I think this nicely shows the visual nature of FISH. The image here shows a slide with trisomy 21. You'll see that two chromosome-specific probes are used. The green probe anneals to unique regions of chromosome 13, whereas the red probe is specific to chromosome 21. And on this slide, you'll see that there are 3 red signals, whereas there are only 2 green signals. So the 3 red signals are indicative of there being trisomy 21 or Down syndrome. Common microdeletion syndromes with consistent breakpoints usually arise due to non-homologous recombination. And there are various commercially produced probes for these different microdeletion syndromes. So here we have an image showing DiGeorge or velocardiofacial syndrome caused by a deletion at 22q11. The green probe is a satellite probe identifying chromosome 22, and the red probe is specific for the commonly deleted region. The normal image on the left shows 2 red signals for the 22q11 region, and the image on the right shows a single red signal for the 22q11 probe, indicating the presence of a deletion. And this one on the right can be seen in both the interphase and the metaphase nucleus. But one of the main uses for FISH is in the confirmation of array-CGH finding. And this shows a 1q21 deletion, which has been picked up on array-CGH. And that's shown on the left-hand side of this slide, where you have the ideogram, and then next to it a graph with a peak representing the 1q21 deletion. So to replicate the finding, a suitable FISH probe has been chosen, and the FISH image here on the right shows a single red signal representing the 1q21 region, and therefore confirming the result. FISH can also be used in the identification of marker chromosomes. And the karyotype here shows an extra piece of chromosome material, labelled here as MAR, standing for marker, and the suspicion looking at the karyotype was that the marker came from chromosome 2. So FISH was used to confirm this, and the image on the left shows whole chromosome painting, revealing that the marker chromosome is derived from chromosome 2, and that's painted red. And the right-hand image shows a green alpha satellite probe for chromosome 2, again confirming its origin. And as mentioned previously, FISH is now widely used in hemato-oncology, and that's to aid diagnosis, estimate prognosis, and guide appropriate drug therapy. And the image shows the use of FISH in the diagnosis of chronic myeloid leukemia, or CML. Probes to BCR and ABL regions are used, and this shows the classic translocation between chromosomes 9 and 22, and this is known as the Philadelphia chromosome, which is pathognomic of CML. The arrows point to the translocated chromosome, where the red and green signals can be seen to bind side by side. So having thought a little bit about how FISH works and then the clinical applications of FISH, I'd like you just to take a little bit of time to think about both the advantages and the limitations of the technique. And there's some questions for you to do that will involve just a little bit of work online. But I'll be very pleased if you could share your findings with us, your educators, and also your fellow learners.